We are live. Welcome to episode number 1 of Secrets of Success with Dr. Shane Needham. And we have the theme in this podcast of never be outworked. And I couldn't have a more perfect guest today, and Dr. Chad Briscoe. We'll talk about our stories a little bit. Since this is episode 1, I'd like to introduce you to this podcast, to myself, tell you a little bit about myself and where you can see more about me and more about the podcast and more about streaming and asking questions. We'll get into that as we go. And so I'm Shane Needham, and welcome to this podcast. And I really like to consider myself, I've never wanted to consider myself as one identity. I'm Shane Needham. I'm not necessarily just a scientist. I'm not just an entrepreneur. I'm not just a disciple. I'm not just a father. So I'm many different facets of that, and that makes me who I am, and we're all that way. And we're going to be talking a lot about that as we go on through these episodes. And we have great guests coming up. Today, we're going to be talking about COVID-19. In a couple of days, we're going to be talking to a pastor online, and we'll have that live, and I'll give you more information with that about faith through these trying times that we're in right now. We're all quarantined, where most of us are. We're self-isolated, COVID-19, there's fear, there should be, and we hope to quell some of those fears, or at least answer some of your questions about the testing today. And so, who am I? I'm a disciple, I'm a father, I'm a scientist, I'm an entrepreneur, I'm a bodybuilder, I'm a power lifter, and I'm most proud of being a father, by the way. I'm also, the original co-founder of Alturas Analytics, and as a chief scientist, and this is our 20th year of business, and I've known Chad, I think, for 21 years, so we go back a long, long time, and so that's my story. You can actually go and check out my website. I'm glad to call myself a podcaster now as well. As things have happened in my life, and I guess I've just got more experience, I've also become a mentor, and that's really important to me. And I want to say, with this first episode of podcast, we already have so many guests lined up. Thank you to all of those people that have reached out and want to tell your story, and you have something to add. We have professors, we have pastors, we have economic advisors going to be talking about what the economy is going to happen to the economy with COVID-19 and what has happened. And so go check out my website at drsneedham.com. Again, drsneedham.com. You can check out also my TED Talk there. I gave a TED Talk last year about this time, about never being outworked. And so that's kind of been just my motto. It's something that my dad taught me. And so I'm super excited about this. I have a PhD in chemistry and also a bachelor's degree in chemistry as well. And so that's kind of how I met Chad. And so we're just going to be very organic here. Now, we're in challenging times. There's self-isolation. A lot of people are out of jobs. A lot of people don't know where their economics and successes will come from. And, I want to be a part of that solution. So reach out to us. We're live on Facebook right now. Go to my Facebook page, Shane Needham. You can find me there and ask questions there. And remember, the 10th comment or question, not from the same person, don't do 10 questions all at once, is going to earn a $50 gift certificate to the restaurant of their choice in their community. And we have watchers right now, I know of this, all around the country, in the United States, in New Jersey, in Kansas City, in California. We have people all the way from Brazil and Europe that are watching right now. This is really, really cool. And one of the things with this podcast as a scientist is, as you know, with most scientific information, as scientists, we publish in what we call white papers or journal articles. And then that can take, Chad, what would you say an average journal article takes to actually start and write and get published?
Oh boy, you're talking a year to 18 months if you're doing well.
Exactly.
And that's if you can really be focused on that writing. Yeah.
And that's why I wanted to start this podcast, is because this is 2020 and people are looking for immediate information. And so we're here to give a voice from scientists to the communities that are non-scientists, and we're going to try to use layman terms to try to help explain sometimes what the scientists do. And so Chad and I are both bioanalytical scientists. And so what we do is we are involved in testing drugs and metabolites and biomarkers and things in and out of the body externally. I'm just going to try to explain it very easily. And we develop analytical tests using mass spectrometers and high performance liquid chromatography instruments and all these things that you see on CSI and stuff, but we really use them. They don't. So anyway, so that's what we want to really do. And so I want Chad to kind of tell the story first of how we met, what his memories are, and then I'll say what my memories are too, because I they might be the same, they might be a little bit different. So I have Dr. Chad Briscoe. He is on the first episode, and this guy, when we met, we were in our 20s. I know that, and just had more hair or less gray hair, all those kind of
A
things.
lot more hair.
Yeah, and I just knew that this guy was going to be a rising star. And we just immediately connected. I'll let him tell a story, but he had that never be out work ethic. And those are the people that I want to interview and be able to tell their stories, and they can tell their stories. And so Chad has gone from being in this bioanalytical industry. He also has a PhD in chemistry. That's also a never be out work story, very similar to mine, actually, working and doing your PhD at the same time, working full time and doing your PhD. And so, he's been in this industry for 20 years. He's known as one of the global thought leaders. And when the guy gives presentations, when the guy writes publications, everybody knows that it's transparent, it's real, and you're getting good information there. And so that's why I wanted him as my first guest. And it's just very timely. So Dr. Briscoe, I call him Chad. Chad, tell us about yourself, and then tell us about, our stories together and how we met. And then we can get into more details.
Yeah, I really appreciate that, Shane. And it's really it is an honor to be your first guest. I think we figured out we were going to do this in Kansas City when you came out to Kansas City, and we were having way too much barbecue at Joe's. So,
it was perfect.
Yeah, that's a great kind of planning meeting. But so, as you said, I've been in the bioanalytical industry for many years. For those of you on the podcast who don't know this area of the pharmaceutical industry, bioanalytical is it's really testing for drugs and biomarkers and other types of compounds that are in blood, plasma, human tissue, basically anything that can come from the body, any part of the body. We'll measure drugs in that space. So that's where Shane and I come from. That's part of the industry. So that's where my expertise that I can offer as far as the COVID-19 space, and then we'll talk a little bit more.
So Chad and I are probably going to have to catch each other on this, because we usually talk to scientists. We talk to scientists that are in a group of hundreds or 1,000 in a room, and we're giving presentations, and everybody knows what we're talking about. So Chad, I know it might seem rudimentary, and that is not offensive to anybody, because we want to make sure that scientific information gets out to people like our moms that may not know about science, our grandmas, our aunts, our dads, our uncles, our brothers, our sisters. Can you describe what a biomarker is? Because I think that's important, even when we start getting into COVID-19 testing.
Yeah, that is actually really, really important related to the COVID-19 testing. So we do two things. Again, as a bioanalytical scientist, we'll measure, like I said, we'll measure any kind of compound in the blood or in tissues of a human, and some of that might be the drug that you've taken. If you take a drug, we might measure that. So if you're taking aspirin, then we'll measure the concentration of aspirin, but that aspirin is going to do something to you. So in the case of aspirin, it's pretty simple. It might reduce your fever. And so your temperature would be an example of a biomarker, but there's other things that might happen in your body as well. Some drugs will cause you to have an allergic reaction or an immune reaction, which could be the case with some of the COVID-19 treatments. And so we'll have to measure other compounds in your body, in your blood, in your tissue, to show that you're having a reaction to that. And that can be something that's good, and you want to look for that when you're developing drugs. You want to see an action. And some of that can be bad. It can be a way to measure side effects. So we can use it for both of those approaches. So yeah.
And when Chad talks about compounds, what we mean there is it's a chemical structure in the body that we can detect and analyze and quantify, tell you how much is there and what it is in the body. And so compounds are just another name for chemical structures or molecular structures.
Yep.
And so it's just kind of a common term we use in the industry. So Chad, that was great. Tell us a little bit about you want to talk about our story and how we
I want to talk about our story. Yeah.
Okay, let's do that.
I moved to Lincoln, Nebraska in 1997 and was working for a bioanalytical company in Nebraska. And one of the things I was looking for, I had a master's degree at the time, and one of the things I was looking for was some sort of professional development. And I had decided that I was thinking about maybe I'll go get a PhD. And also at the time I had one kid. I had another kid that was on the way, and so it just didn't seem like it made sense to quit work and go back and get a PhD. That just didn't seem financially sensible. And I went to one of our major national meetings that Shane and I have been to many times, the American Society for Mass Spectrometry. We won't get into those terms, but that's the name of the meeting for the science folks here. And Shane was presenting a poster that I was interested in, just the science. I didn't know Shane at all, and he was working at Pfizer at the time, and so usually Pfizer's doing great presentations. So I went over and looked at his poster and was interested in the science. Really cool stuff. And he told me his story, and he said, "Well, yeah, I've been doing this PhD for a while. I've been doing a PhD while I work full time at Pfizer, and I work in the evenings and on the weekends and things like that." And I said, "Wow, that's exactly what I want to do." And I had previously been a bit overwhelmed with even thinking about doing it. I'd seen my brother go through a PhD, really firsthand, and how much work that is, and so I really appreciated, what the challenge was in front of me. And so I have to say, Shane, you really you inspired me. I mean, I was pretty sure I was going to do it at that point, but that was where I said, "Yep, I can definitely do this. It's definitely something that can be done." And so I did it. So yeah, so that was the first time we met. I don't know when we met again. I don't remember the second meeting, but
I don't either.
yeah,
Yeah, I think, was that 99 or 2000?
I it might have been 2000. Yeah, if it was
Yeah.
2000, I was already a year into it then. Yeah,
Okay. All
the way
right.
I remember, but it might have been 99. It might have
But
been 2000. You presented your penafluorophenyl chromatography count.
2000. That was 2000.
2000. Okay. Okay. We won't
Yeah.
explain what that is either, but
Yeah. Yeah.
yeah.
PP stationary phases. That was
Yeah.
actually my
Yeah.
dissertation. And
Yeah.
so,
Yep.
and it really helped in the bioelectrical industry and when I was
Yeah,
doing work at
absolutely.
Pfizer
Yep.
at the time as
Yep.
well. So,
Yeah.
and I think we might not have met it again until the Midwest meeting at Land O'Lakes. I
Yeah,
mean, face to
probably.
face. I
Yeah.
mean, of course I
Like
saw
three,
your
4 years
name.
later, maybe. Yeah.
Yeah.
Yep.
So
Yep.
what a
Yeah.
great story. And his story was similar to mine in that, having a family, working full time, not wanting to do the traditional PhD
Yeah.
graduate school thing where, you don't get job experience. You don't make a lot of money. You're not, can't save for a 401k or anything like that. And so I chose the way that was probably more work, and I would do it all over again. I really,
I'll tell you what else we both did, Shane, is we both went pretty much the traditional route with the PhD. We didn't do a mail order PhD. We didn't skip the steps, and
no,
that's something else that I think is unique about what we both did. And I'm really proud of that because I did it. I would say I did it the real way, right? I feel like I think you're Rhode Island and I was Nebraska. I think we have legit, right? I mean, I feel like we have
absolutely.
legit PhDs from two
I
great schools. So.
Yeah, and I was just thankful for my my supervisor at work, my PhD
Hmm. Yeah.
advisor, and all the
Yeah.
people that supported me during that time. It was really
Yep.
a lot of work, but it was not, it was, and it learned a lot through the process,
Yeah.
and so
Yeah.
it was
Absolutely.
really, really good. And so Chad and I have been, colleagues on and off, and met each other on and off at meetings ever since for the last 20 years. And our careers have obviously grown and diverged a little bit and converged a lot. So
Yeah. Yeah. Yeah.
it's a really, really cool thing to have you on as our first guest, Chad. So really, really appreciate it. So
You
Chad,
bet.
can you talk a little bit about what the bioanalytical industry does, some more, a little bit,
Sure.
and how it's changed in the last 20 years? Let's talk about
Yeah.
that. And
Yeah.
maybe let's segue that as you complete that into how that relates to COVID-19 testing. Because
Hmm.
here's how I'm gonna open this up. COVID-19 testing might not have been possible 20 years ago, or it would have been a lot
Would have been
different
a
and a lot
lot
longer,
different.
a
A
lot
lot
different.
different.
And
Yeah.
we'll talk about those details, but this industry is changing rapidly, rapidly.
Hmm.
So Chad, talk about the industry, talk how it's changed in the last 20 years.
Yeah, so we've certainly seen a lot of changes, and I think when Shane, we have talked about writing a paper about this, kind of what we've seen, how we've seen the industry change, and we both walked into it, I would say, almost in phase two, where it had, just past the birth. Maybe the industry was a toddler or something at the time when when we came into it, but when we came into it, it was very much assaying for chemical structures, so what we would call small molecules when we talk about those compounds. So drugs like ibuprofen and aspirin and morphine and drugs like that. So these types of molecules that the more traditional types and kind of not pretty early in our careers there in the early 2000s, there were a lot of regulatory challenges that we ran into, and so that marked a lot of the early part of what I was learning and what I got involved in. It was kind of a slow evolution, but meanwhile in the background there was this biotech boom coming along that and I was very focused on the chemical aspects of it in my part of the industry. And I think things like sequencing the human genome that brought so much to it. And I'm trying to remember exactly the years when that was done, but
I
that
think that was about, wasn't that Venture and Craig Venture and
yeah, Craig Venter
yeah,
in right around 2000, right?
2000. I think it was
That
2000.
happened. So
Yeah.
so that that really drove a huge boom in the biotech, and then as you got closer to 2010, that was when you really started seeing a lot of these biotech drugs. Amgen was already huge. You saw Genentech getting huge. A lot of these other companies. You saw other companies like Bristol Myers Squibb that would change their portfolio from being all traditional small molecules to being at least half or more biologics. And so a lot of us
So
have had to
Chad,
change and shift,
I'm gonna
right?
I'm gonna
Yeah,
catch you on
yeah,
that. So
yep,
remember your thought.
yeah,
Again, the majority of our audience, I hope, are non-scientists. That's what
yeah,
I hope.
yeah,
I have thousands of friends on Facebook, and I'm sure you do too. And a lot of them don't know science. So describe a biologic, and
yeah, yeah.
describe, because
Thanks. So,
we.
yep,
So I want to segue this way. We see a lot of advertisements on TV. For example, I'll talk about Humira. Okay, that's a
mm-hmm,
biologic.
yep,
The layperson probably doesn't know that. So
right,
describe what's a
yeah,
biologic and
yeah,
how it's maybe different than a small molecule,
yeah. So
like a
that's yeah,
traditional, like a
yep,
traditional aspirin or ibuprofen or something like that.
yeah, sure, sure, yeah. That's I'll do that. So those traditional, when we talk about small molecules, to us, and again we think about molecular weight, right? So what's the weight of all the atoms that make up a molecule? So the biggest difference between typical small molecules, they may have a molecular weight of around 300, right? So you might not, some of the listeners might not have a perspective on how big 300 is, but if you think about a large molecule or a biologic, it's probably going to be around 180,000, right? That would be something like Humira, right? Like an antibody. So some of them maybe are 30,000. There's everything in between, but typically when we talk about those biologics, we're pardon me, we're thinking of something around 180,000 molecular weight. So if you just think about it, something that's that much bigger, what is that, 600 times bigger, then you need different ways to analyze those drugs. You still need to analyze them. You still need to get the same types of data, but you need new ways to analyze them. So along with that's a great segue into starting to get towards this COVID-19, because along with the analysis and developing the tools to analyze those large molecules, those 180,000 molecular weight, you needed new technologies based more on biological mechanisms for detection versus physical and physics. So Shane and I, our background, our training is more chemistry and I would say physics than it is biology. And I took a number of biology classes. Shane probably did too. But but as this industry has shifted, one of the directions I've gone in my career is I'm doing much more different types of analysis. So we talked about the biomarkers. I've gotten very involved in the biomarker space, analysis of these huge biologic drugs using these different technologies. I won't explain what an immunoassay is, but for those of you who know, having to learn about immunoassays and this type of technology. analysis of cells and whole cells. Meanwhile, I know, Shane, one of the things you've done is you've said, all right, we're going to use some of our traditional tools.
Yeah.
While I've learned a lot of new tools, you've spent a lot of time taking traditional tools and saying, okay, we can use these same tools and we'll get different types of information, but we'll get, better information in some ways. And then there's drawbacks, right? So there's strengths and drawbacks. I think that's something that I've seen you do a lot in the last 10, 12 years with the
Yeah,
emergence.
that's one thing my company has focused on is using traditional tools to look for some of those larger molecules. And I think especially as we segue into COVID-19, for the non-scientists out there, right now, especially with COVID-19 and all that's going on, and when Chad talks about traditional tools versus current tools versus better information versus worse information, that's the whole thing. It's about, there's really no better or worse. There's just data.
Mm-hmm. Yeah.
And so a traditional tool is going to give you a certain set of data that is not wrong or right.
Mm-hmm.
And then maybe a newer developed tool will give you a certain set of data that is not wrong or right. And that's where scientists have to come in and look at the data, review it, and then make some kind of a hypothesis like, hey, this is what's happening. This is why the data is different or why it's the same.
Right.
And you have to do that for every single new test that you develop.
Mm-hmm.
When you co-develop it, if you have two different methods of measuring it. And that's okay. And we'll talk about that with COVID-19 because there are lots of tests right now. None of them are wrong. We just have to look at the data and know which is the direction to go. So Chad, so keep going on with the conventional tools versus the newer tools.
Yeah. So leveraging those types of tools that were developed when we start talking more about COVID-19, it's because of these tools that we've developed in this journey over the last 10 or 15 or 20 years. I mean, the story really goes back to the 60s and 70s, but really where there's been a ton of scientists involved, and again where this type of testing and these types of assays came into the bioanalytical space has just been in that last 10 to 15 years. So immunoassays that I mentioned, that type of testing that you can do, we're back, I think it was 71 or 72 that the first immunoassays were done.
Chad, can you give an example of immunoassays that the public would know about? I mean, I can think of some, but I want to.
Sure, yeah, I'll tell you, the easiest immunoassay that many of you know about would be a pregnancy test.
Exactly.
So, and we call that a flow-through immunoassay, and it's just a very simple test where when you pee on that stick, your HCG is a human chorionic gonadotropin, right? Which is what you measure with the pregnancy test. It flows up that little stick, and if it sticks to a certain spot, then you get a pink line, and if it binds to two sticks, then you're pregnant. One is a control. You get one line, and two lines means you're either really happy or really nervous. Depends on the result you expected.
Absolutely. Yeah. Okay. So that's good. So move forward.
Yeah. So moving forward, this biotech revolution of different types of drugs being developed and things like that. It wasn't just these antibodies and these proteins, right? These giant molecules. That's the name for some of these giant molecules that we learned how to analyze. But then we also, again, we were going back to that sequencing the human genome and that genetic studies and being able to study the genes. And so recently, and this is really just the last 5 to 8 years, we've started to see development in a space called gene therapy and cell therapy, where we are actually manipulating the genetics inside a cell with a lot of different ways that we're doing that. And we're doing that to cure diseases, right? And we can cure a lot of, there's a lot of potential in that space. Cancer is certainly a classic, and there's some other diseases, the so-called SCID disease, which is the boy in the bubble disease, right? Where if you're born essentially without an immune system, that's something that looks like we're very close to a cure. Actually, we have a cure, but the problem is there's side effects, right? So it's kind of figuring out those side effects. And that's where, when we talked about the biomarkers, going back to those biomarkers, that's what we measure in that case. There was a lot of a lot of biomarkers that cause an issue. And for the scientists that are on the call, that's that cytokine release. It's that reaction that you need to measure, that you need to track, that you need to try and minimize that sort of an effect. So these tools that have been developed over the last 20 years, 20 plus years, have really culminated in coming into the hands of bioanalytical scientists, which, as we talked about, that's where we measure things in biological fluids. And we've built, as a bioanalytical community, people who are experts in testing. Now we're experts at testing with immunoassays to measure these large biologic drugs, which is part of the COVID-19 reaction. And we've brought these genetic testing tools into play. And because the last 5 to 8 years, we've become, we, not necessarily Shane and I, as much as people in our industry and people doing this measurement for our application of testing, have become experts in this type of measurement. That is also very critical to the testing that's going on for coronavirus. So that kind of brings it all together, right? The industry has come together, and the right types of testing have come together. And that's where I think we're at a great spot to have the know-how to do the testing. There's no doubt that we know the right ways to do it. It's a question of how quickly can we get the tests, evaluating the trade-offs of the quality of the test, things like that.
And the price. I always look at it.
And the price.
Yeah. So I always look at anything available to a consumer as a triangle. And there's price, there's quality, and there's speed. And in general, I heard this a long time ago, you can get 2 to 3. You can't have them all. You can't have them all.
Yep.
And so, a really good example of a ever-changing test, that's an immunoassay that Chad talked about. They're trying to maybe take some mass spectrometry assays and change that and kind of at least confirm the data, is like PSA testing. So PSA testing, the data has been all over the map because a lot of it has to do with the test and how inaccurate it is. Do you know much about that test, Chad?
I'm guilty of reading some of the comments on Facebook.
Oh, that's okay too, because I didn't want to do it. So if you see anything, I need to. So hey, that's a good point.
I did. Say it again. Say it again.
Okay, so if you guys,
classic teleconference mistake.
Yeah, it's all right. If you want to comment on Facebook, if you want a question, I'm not going to guarantee that we can get to it live, but Chad and I will do our best to get to those questions later.
Yeah.
But the PSA test, that test, and what would I say, the healthy range has been changing for the last 20 years. And now there's possibly many reasons for that. One of the reasons is that, I don't want to use direct terms here, but the test has been traditionally highly inaccurate and unreliable. And so, and unfortunately, and the same with some of the tests that you can use for mammograms and some of the chemical biomarkers with those. And unfortunately, then sometimes surgeries happen, and because of a chemical test or some data, and that patient is never the same.
Yeah.
And so some of the science that Chad and I are involved in as bioanalytical and clinical science kind of converge, I won't get into too much of that, is making sure those tests are accurate, because right now they're not. And patients' lives are dramatically negatively changed because of some of those inaccurate results. And so would you agree with the PSA test on that one, Chad?
Oh, yeah. In fact, I just spoke with a customer not too long ago that is actually looking at a prostate cancer drug, and we were looking at better ways to measure measure their success of that prostate cancer drug. And they said, well, there's PSA, but it doesn't really give us what we want, but there's not really, so one of the things that we're doing for them is looking at some really novel ways to look at some different reactions, in this case, for the drug. And in this case, actually, it was a gene therapy. It's one of these new gene therapy drugs that I talked about that they're working on, but they're really struggling with anything to measure it short of being able to cut out, right? If they can cut the whole tumor out and then they can and then they can look at it, that can tell you something. But that doesn't really work with early phase trials, right? People don't want that invasive surgery, and it's not always warranted. So
yeah. So that's a great example. So that's what I'm excited about. I'm excited that my company's involved in this industry and Chad's involved in this industry where really we're trying to treat disease. We're trying to cure disease. And there are some great success stories out there. And there are also sometimes you read the media and yeah, there are some horror stories as well. But I want you to know, as scientists, we have the best intentions, and the people we work with, our customers and our labs, we have the best intentions, and we're always trying to do what is best for the patient in this ever-changing world of sometimes people don't treat themselves right, but we still have to treat them. We still have to treat them. We want to treat them. We want to be a part of that community that is actually successfully treating people. And so that's part of our job. And I love that about it. It's very inspiring to me to be a part of that cure. And so Chad, let's get into COVID-19 testing.
yeah,
So
yeah,
Chad was using some terms on me when we first started talking about this, and they even went over my head. So I'm going to learn at this too. And I always learn every time he speaks. And so I'm super excited about it. So let's just start with giving a quick overview, and then I'll ask maybe
sure.
some more specific questions that other people have already asked me to say, hey, can you ask your guest this? So go on.
Cool. So I think six, if you look back maybe 6 weeks or even January, right? Even go back to January when I was in China and you start hearing about testing. What people want to know is, are people sick, right? And so that's the first thing you want to know. Does somebody have this, right? Somebody have this, can I catch it? So we started hearing about some testing for it, and there was, I think the first test came out of Berlin, and then there was a WHO test, and then the CDC test, and the famous failed CDC test. But that happens also sometimes. So I don't know the story on what happened there, so I don't judge. We certainly had failed assays, both of us in the past too, more than we'd like to count. So let's not judge there. Nobody, But anyways, those early tests that you heard about, were all based on a technology called qPCR. So we won't go into what qPCR is. If somebody wants to message me and ask, I can send them some resources. But qPCR is measuring the RNA. So very quick lesson on how viruses work. If you haven't seen a good YouTube video yet through all this, I think a lot of people have seen it, but just a reminder, your virus is like a tennis ball, and it's got these spike proteins on the outside of it. That tennis ball is filled with genetic material called RNA. RNA is like the cousin of DNA. That tennis ball then sticks to a cell. It loves to stick to cells in your lungs. And that's why it's a lung disease. So it sticks, it opens up, and it basically deposits that RNA material, that genetic material, inside your cells. And then it tricks your body by dumping that material in there. It tricks your body into making more virus particles. So the RNA gets replicated into more RNA. And the other RNA gets turned into proteins, which proteins are like the for the RNA for the virus. The proteins are basically the yellow fuzzy stuff that's the outside of the ball. So basically, your cells, your own cells, are tricked into making more of the virus itself, and then your own cells become factories for that virus. So that's how the virus works. And so if you want to see if somebody's really sick, you're going to be measuring the RNA. So those early tests use that technology, that qPCR, which, by the way, when we talked about the genetic, sorry, you told me to turn off all my notifications. I just got a phone call on my watch.
Hey, you know what? In these times, we totally understand, Chad.
I take my watch off. But anyways, so when we talk about these RNA, measuring the RNA, that's the same approach that Craig Venter used, essentially, right? It's a more sophisticated approach, but very similar approach to what Craig Venter used when they were sequencing the human genome. So it's just an evolved version of that.
And so, Chad, when you're measuring the RNA, are you talking about measuring the RNA from the actual virus?
Yeah, well, you're measuring the RNA from the virus, or the RNA more so that's been replicated by the cell itself. Yeah.
Okay, and I know we don't want to get into too much. We don't want to get into too much of the detail of that because that can be confusing.
Hmm.
But what he just said there is important. Measuring the virus directly versus how it's some of it that's
Right.
replicating. Those are
Right.
two totally different assays.
Yep. Yep.
Which one is correct? We don't know.
Yeah.
Which one is actually going to define if you really have a late stage disease or you will have?
Hmm.
We don't know.
Yeah. Yeah.
Which one's the best test? We don't know. That is information as scientists that we're still collecting, and we might not know for a long time. A
Right,
long
and
time.
you can imagine that measuring that RNA, your DNA is obviously it's tiny, right, on those terms, but the piece of RNA that you're going to measure is a tiny piece of the overall genetic material within that cell, and so that's where you talked about the accuracy of the assays, and that's where some of the challenges come in, right? How accurate is the assay? It's pretty unlikely. So there's two categories that are really important in this testing. There's the false negatives and there's the false positives, right? So you can imagine a false negative means that you actually have the infection, but your test comes back and says you don't. And a false positive means you don't have the infection and your test comes back and says you do. So I was looking for some data this morning, trying to kind of see what what they think is the false negative, false positive rate. The false negative rate is quite low. Or I'm sorry, it's the false positive rate is quite low. I'm not sure exactly where that's at, but it's pretty hard to get a positive reading if there's nothing there to measure, right? That's probably a contamination. That's probably a lab error if you get a false positive, but a false negative is pretty dangerous, because then you can get a negative result, think you're not sick, and your doctor sends you home untreated because they think you have something else. So
Unquarantined.
yeah, exactly,
Yeah.
exactly.
And
So
so what did you happen to see any data on that with the
so
data?
yeah, so the false negative I saw anywhere from 5 to 30%, and
That's a
it's
lot.
30% is huge.
Yeah.
30% is huge. So yeah, so 5% is still way too much, but it's probably an acceptable level. But yeah, that going up to 30% is really scary for that false negative. That, yeah, again, that means that you might think you have it. You go in, you get a test, and you say, oh, maybe I just have the flu, and then you maybe go around and say, well, I just got the sniffles. I'm going to go out grocery shopping, or feel like I can go see my grandma because I just got a test and I know I don't have it, so I'm safe. And then you go and you give it to her, and you spread that. So I wouldn't want anybody to necessarily take healthcare advice from me. But what I read is that some doctors are saying, hey, if you think you have the symptoms, and whether you that's me, whether you test for it or not, you whether you test for it or not, you should act like you have it, right? Or at least quarantine for another 72 hours, pretty severely.
Yeah. And
Yep.
Chad talks about that, and I think that's why we have to be really
Yep.
careful. I know that's this is a very timely topic. I'm glad I've got Chad on the podcast. Testing, testing, testing. Everybody's, where are all these millions of tests? Well, Congress or the president or the WHO says we have
Mhm.
millions of tests. How come people aren't getting them? Well, these might be some of the reasons. We don't know. We
Right.
don't know, but
Right.
maybe they figured out, they found these, they're like, oh, wow, these are wrong 30% of the time. That can be
Mhm.
really dangerous.
Yep.
And so
Yep.
it makes no sense economically, scientifically, healthcare wise, to get
Right.
a test out there that is not reliable. And so I know the communities are asking for them. I want accurate and reliable tests
Yeah.
for COVID-19, but it's just not that easy. And
Yeah.
these tests, by the way, can take months to really develop. By the time you get reagents and all these kind of things, and make sure there's accuracy and precision and all these things that we won't get into the details, but to know it's specific and all that. So we've heard about false positives and false negatives. Chad, we have heard a
Mhm.
little bit in the media when you dig a little bit more into the science of
Mhm.
COVID-19 testing about,
Yeah.
is the assay specific? What is
Right. Right.
describe specific?
Yeah, so I kind of touched on that a little bit. So you can imagine when you have a piece of RNA, and a lot of us might remember from school that RNA is only made up of 4 different compounds. So if you have a string of 4 different compounds that repeat themselves a billion times, and you're only going to measure a small piece of that, maybe 1,000 pieces or even smaller, that your test might not recognize that perfectly, right? And so if your test recognizes it perfectly, then you're perfectly specific. If your test gets it wrong sometimes and thinks that a different piece is the piece of RNA, then that means that it's not specific. So another way to look at that is, let's say we talked about measuring acetaminophen and ibuprofen, right? So if you were measuring acetaminophen and ibuprofen and you couldn't tell one from the other, that's kind of selectivity too. But if you can tell one from the other, then
We won't get into the IUPAC.
yeah, then that would be, that would also be a challenge with that test, right? So that's
Well,
something we
I
always
think,
test for when we develop assays.
well, and Chad brings up very good points, and I think it's important to kind of let the audience know that when you're doing tests for, he talked about it earlier, large molecules or biologics or biological entities. And that's basically, yes, our body does have small molecules in them as well, lots of different things and hormones and so on and so forth that are small molecules. But these large molecules that we're testing that Chad was talking about can be 50,000 AMU molecular weight or 100,000, 180,000 or whatever. We rarely, would you agree with this, Chad? Well, actually, I don't use the word never as a scientist very often, but we rarely detect the entire molecule. We detect pieces of it.
Right.
And
Yep.
and we call that like a surrogate test. Like,
Right.
well, we
Mhm.
think this part of the molecule is very
Yep.
specific for the rest of the molecule. Well,
Yeah.
here's what it could be like, okay? Let's just think about this. It's like me cutting off my arm and
Hmm.
go and give it to somebody, and now somebody has to say, "Oh, that arm is Shane Needham." It's not the same.
Or
It's not,
just your fingerprint.
or
We
your
can just put your
fingerprint.
fingerprint.
Yeah.
No, that's Shane Needham, right? Yeah.
Exactly.
Yeah.
And
Yeah. Yeah.
things like that, it's just not as easy as we can think, even though like with fingerprint technology, we're pretty good now because we've had years to develop it. And
Hmm.
so, but this is all new. Detecting viruses, detecting RNA, detecting
Yeah.
DNA. I mean, you got to
Yeah.
think Watson and Crick kind of really discovered RNA, DNA, so on and so forth, and double helix. 57, they won the Nobel Prize for that.
Yeah. 57 or 58, I was thinking. Yep. Yep. Yep.
I had the honor to actually see one of them actually give a talk, one of their last talks before they passed away in the mid 90s at
Cool.
either
Cool.
Harvard or
Yeah.
MIT.
Yeah.
It was really cool.
Yeah. Those are
It,
the things scientists get really jazzed about.
it was really cool. Yeah.
Yeah.
And so, but we're just detecting pieces of this and we just assume that it's going to be the whole molecule, and it might not be. And so that's what he's talking about with
Yeah.
specificity, and it gets into the reliability of the test too, is that if it's not specific, then is it reliable? And he's already talked about some of those numbers. And so um that's direct testing. And so Chad, you want to kind of wrap yourself around that before we get into more of antibody type testing, and we'll get
Yeah.
to that as
Yeah.
well.
Yeah.
So that
So
what Chad's talking about right now is directly testing if you have COVID-19, if that virus is in your body. That's not the only way to test if you
right.
have this
Right.
disease. Okay.
And
So go
yeah.
ahead, wrap it up,
Yeah,
and then
I
we'll
think
get into the next one.
yep. I think the last thing I was going to say on that, and you touched on it also, is that these tests all have trade-offs, right? And as we advance the technology further, we're going to get better and better at having better rapid tests that are more accurate, that have better specificity, that have fewer false negatives and fewer false positives, and we'll get there. But bringing out these tests super fast, we need it, and we need to keep doing it. It's kind of like real time, put stuff into action here where we can. But I saw a 5-minute test, which is amazing. It actually uses the same tool as the rapid. I think it's the same as the rapid strep test, or maybe it's the rapid flu test. But uses that same instrument, so they can basically, it's a flow through technology, and so I don't know the accuracy. I don't, I'm sure it's pretty good. I'm sure it's good enough to detect, but I'm sure that in a year we're going to have a lot better 5-minute test available, right? So right now it's like, let's balance cost and timeliness and getting these tests out there. So that's the, that I think is a challenge. Like you said, it's that three-legged milking stool model, right? Cost, quality, and timeliness. So
choose
you
choose two of the
can't
choose
choose
two of the
two.
three.
Yeah, pick two. So
I mean, for example,
yep,
a PSA test is really inexpensive, but if we get it to be really accurate, it's going to be 4 or 5 times the
yep,
cost. Are
yeah,
people going to want to pay for that? We
yeah,
don't know.
yep, yep,
Does insurance want to pay for that? Would the government pay for that? Those are all
mhm,
questions that we have to ask all these questions when we, as scientists, when we develop these tests, is it economically feasible? And so,
right.
no matter who's paying for it, government, yourself,
Hmm.
an insurance company, is it economically feasible? Right.
Right,
So
right.
Chad's talked about the direct testing, which
Hmm.
that absolutely has its pros, it has its advantages. Now, what about indirect testing, Chad, where you're testing to see if you have antibodies
Yeah,
to that particular
yeah,
virus, and then
yep,
kind of um, compare those two, and what are the
yeah,
advantages and disadvantages of each?
yeah. So this is where it gets really interesting and
It
also
is. He already
a
told me
little
this before.
bit
It's really
more
cool.
complicated, right? So I think probably it's pretty commonly known that when your body is immune to something, you have antibodies against it, right? So you get a flu vaccine to create antibodies. Your body, when you all the time you're taking in pathogens and you're building up antibodies. When we think about as kids, you say, oh, if my kid eats dirt, and gets something, now they'll build up an immune response. They'll build antibodies in their body, right? You get these things called memory B cells that basically they hold the antibodies that remember how to fight off infections. So when you get invaded by this vaccine, one of the ways that your body is going to fight it, there's a few ways that it does it, but one of the main ways, probably the main way that it's ultimately going to eliminate it, is by generation of antibodies. So those antibodies are going to grab onto the
The virus.
virus and grab onto that virus and tell your other cells that this is foreign, you need to destroy it. So they kind of, your body figures out that it needs to create these antibodies, and then it figures out uh, how to generate more of those antibodies, and then it recognizes those, and your other cells come in, your other white cells. There's special kinds of white blood cells that come in, and they attack it and they destroy that virus. So that's ultimately what's happening when people are recovering from this. It's just,
And that's called it when something that, I guess when something foreign is in your body,
yeah,
like a virus or something, it's called an antigen.
right,
Yeah.
yep,
And then
yeah,
the
exactly,
antibodies develop around it to try to, I guess, excrete it is a better,
yeah,
or just try to
yeah,
treat it, or to it's your body's way of saying, hey, we got to fight this foreign concept that's in
yeah,
our body.
yeah. To tell your body to chew it up and destroy it, as
Yeah.
opposed to internalizing it and letting it take advantage of your nice cells. It brings the mean cells
Yeah.
over.
Yeah.
The mean
Exactly.
cells come over and they gang up on it and beat it up. So
Yeah.
so around, I think it's around 8 to 12 days. There's a few different kinds of antibodies. I don't think we don't need to get into that today, but there's a few kinds of antibodies. So in the neighborhood of, I'll say 5 to 15 days as you progress in the disease, you'll do a term that you might start hearing called seroconvert. So I just want to throw it out there because what that means is your body starts producing antibodies. So if in the next few weeks, I think you're going to hear more and more about this antigen testing, serology testing, antibody testing, and so seroconvert is when when your body starts learning and it starts making antibodies against that virus to eliminate it. And so those antibodies, some of them come in early and they help to fight early. Some of them come in a bit later, and the ones that end up coming in later are the ones that really clean it up and clear it out. You can almost think of it like the Marines land the beach, and then the Army comes in behind them and sort of grunts in and clears off the rest of the island or something. I mean, that's
Great analogy. You
yeah,
forgot
that's
the
kind of
Navy
how
and
it is.
Air Force, though. They do
So,
that before they get there as well. So
yeah, you need the Air Force as well. Yeah,
yeah.
that's but anyway, so these the antibodies are there, and the antibodies, as I said, they are what stick around in your body, and they become part of the memory B cells, and that's what your body would excrete for for a long for the long time. And so these one specific kinds of antibodies, IgG, is what they're called. The IgG, they stick around for a long time. Now we don't know how long they stick around with coronavirus. That's a little bit unknown still right now, but we want to continue to measure these antibodies for for a while. So you can imagine, why is that important? Well, if you want to know if somebody's sick, you measure the RNA. Remember the early testing, you tell if they're sick. If you want to tell if they're recovered, then and if they had it before, you need to measure the antibodies. Now there's nuances in there, right? Of when do you measure? If you measure early, do you detect one? Do you detect the other? It gets, like I said, it gets complicated, but you certainly want to detect the antibodies. That becomes really important. So one of the really important reasons why you want to detect those antibodies as well is, you can imagine, I've been talking to folks in the clinical trial space about about the, different ways of getting clinical trials restarted. Well, you're going to be nervous to bring somebody in to take an experimental drug if you think that they might have coronavirus. Well, you can take a test and tell that they don't have it actively, right? The RNA test. Or you can take the antibody test, which is just kind of in the process of being developed. That's sort of coming a little bit behind the other test, but you can then tell if they've already had it, right? Or you can imagine in the workplace, in the healthcare segment, if you could test people and know that they already had the virus, aren't those the people you want in New York City right now, fighting, and helping to care for people? If you could do that test. Now, I don't think we've had enough sick people in the U.S. yet. We don't want more, but it's still heading that direction. But you could test, and you could know who's immune, who can care for people, who's sort of safe. Now, we also don't know for sure if your immune response is strong enough that you're protected forever. Maybe you're protected for a short period of time. Maybe something like hepatitis B, you need multiple boosters, right? You need a couple of injections. Tetanus, you need boosters. A lot of these, you need a booster shot. And so that could be how it is with corona. It might not be. We just don't really know. So we know that those antibodies come in. We think that probably a secondary infection, if it happened, would be lighter, because you would have some immune protection, but we can't say no way possible will you get this. That information just isn't out there yet. I sure hope that's the case, but these are things that are going to be evolving over the next weeks, months, years.
Yeah, we this is great information, Chad. We just don't know, and it is likely that a lot of us, the majority of us, again, we don't know, will be exposed to coronavirus. And a lot of
Yep.
us may show zero
Yep.
symptoms, but we have
Yep.
the antibodies, and our body did what it was supposed to do. And we're healthy individuals. We eat right. We exercise. We
Yeah.
do all the things that are right. So our immune responses are good,
Yeah.
and so we can probably fight the coronavirus. But those are all details that we're finding out right now. We
Yeah.
just don't
Yeah.
know. We kind of know who's a little bit susceptible. We kind of have some of that data. We're going to know more over the next couple of
Right.
years
Right.
in white papers, which takes years. But I'm going to have people on my show that when we get that information, I want to have them on here as soon as possible, because
Yeah.
I want
Yeah.
to get that out to the public. And we actually have some people coming up that are going to be talking more about some of that science and in our industry and the clinical industry, and who are professors in academia and so on and so
Yep.
forth. So
Yep.
it's going to be really cool. And so, I hope that answered most of your guys' questions about coronavirus and COVID-19 testing. And I think Chad did a really good job about taking the scientific terms and really condensing them into non-scientific terms, to hopefully for the rest of the community that doesn't follow science. And so again, ask your questions on Facebook. We were so engaged. I'm not surprised. I watch Chad give presentations. When I give presentations, we're never at a loss for words, and that's awesome because it's very organic and very transparent. And but this show will definitely evolve. We'll get your questions online. I'm hoping to also it's being recorded, and so I'm hoping to compress it and have it available on my YouTube channel, and it'll be on my Facebook archived as well. There's one thing I want to focus on in this podcast, and the theme is never be outworked. And I think a lot of people ask me questions like, I just can't believe how much success you've had, and you've done this, and you've accomplished this, and you've accomplished this, you've accomplished this. Well, you know what? And I'll talk about that when I do some monologues and some other things, but most people don't see my failures. Most people don't see the things that I failed at, but that's what has actually created success, is that I don't give up. I just get up from that failure, or it's called a failure. I don't really call it that. It's just a learning experience, and you get up and do better. Chad
Yeah.
was an athlete as well. I was a wrestler and a wrestling coach, and that's just a mindset that you have. But Chad, I want to ask pretty much everybody that comes on this show,
Hmm.
talk about what maybe some would consider a failure. And I want you to know I don't consider very many things a failure about you, but that you turned into a success. Can you think about, and I know I put you on the spot with that one.
Ooh, yeah, that does put me on a spot. Hmm. Well, I mentioned that I moved to Nebraska. Well, I moved out to Nebraska. My wife was from Nebraska, and I moved out there after I left. I left the University of Michigan. I left with a master's degree, right? And for a lot of different reasons, but that's a great. I think that really plays into what we've been talking about, because I left with a master's degree, and I, that was, I didn't finish the business that I came there to start. And make
He
long story
wanted
short,
he wanted the PhD. I mean, he wanted
yeah,
a
I wanted a
PhD.
PhD. Yeah,
Yeah.
I wanted a PhD. That was really important to me, and it made a lot of sense at the time. But I turned that around. I think the spot where I landed in Nebraska, getting into this industry, into contract research, I had no idea what it was till I jumped into it. But all those factors together got me in the right place at the right time. I ended up getting my PhD with an amazing advisor, one, really world-renowned professor that just it was a perfect fit, and it really launched me in my career in that direction, and then we could bond over chromatography, Shane. So
That's absolutely right. Yeah, I love chromatography. I've always loved chromatography, and that's a great story.
yeah.
I mean, a lot of people would take that story and say, "Well, I got a master's degree. That's good enough." And I'm not one to brag about letters behind anybody's name, especially in the current environment that we're in. Is
Hmm.
it's going to come down to people that know how to work.
Yeah.
That
Yeah.
people are, and it always has actually.
Yep.
And so sometimes that's a formal education. A lot of times it's not. A lot of
Yeah.
times it's
Yeah.
just working your tail off. But a lot of people may have just settled in Chad's position there, and I got a master's degree. This is okay. And I mean, having kids and a full-time job and a family to support, and then going back to graduate school, truly
Hmm.
amazing, Chad. And
Yeah.
you are
Yeah.
definitely a sign of success, a model of success for all of us. And you certainly can put the motto behind your name, never be outworked. So thank you
Thanks. I
very
appreciate
much. And
it.
thanks for being the
Thanks,
first guest.
Shane.
Now,
Thanks to everybody who joined us.
yeah, I mean, I can't wait to
Wow.
see the comments. I've been trying
Yeah.
to, I know right now, thank you
Yeah.
everybody. My phone is blowing up, but I'm not looking at it.
Cool.
And I'm
Cool.
getting text messages. I'm getting calls. I never in my right mind dreamed that this would be so
Yeah.
successful. And
Cool.
I'm so excited. I am beyond excited. We have enough guests right now that I have lined up for a long, long time. However, you DM me on LinkedIn, on Instagram, on Facebook, and we will set aside time for you. Okay. We want people, we want to hear their stories. We want the communities to hear their stories. And I want to get you on the air. We're not going to just talk science. We're going to be talking about never be outworked. I'm going to have a professional bodybuilder on my show. I'm going to have trainers on my show. I'm going to have power lifters. I'm going to have professors. I'm going to have a relative of mine who struggles with something. That's all I'm going to say. But you're going to love that edition, because it's going to be authentic and transparent and vulnerable, and it's just going to be really, really cool. We're going to be talking about anxiety during COVID-19 and how to deal with it, especially as you're a parent. How do we talk about these things with our kids when we don't know the answers? We don't. How
Hmm.
do we talk about those things? Chad wanted me to, and I hope I can do this with all my guests.
Yeah.
He wanted to talk about a really important project to him. I already had one of his friends reach out to me and tell me about this project. And then I didn't tell him I heard about it, but he told me about it a little bit this morning, and I've already heard about it. And it sounds really, really cool. and just so much philanthropy involved, so much work effort involved. I just love it. So Chad, tell us about that story and
Yeah.
where people can hear
Thanks.
more about it.
Thanks, Shane. Yeah. So I have a long history with Brazil. I went there first in high school, actually twice as a student, and a good close friend of mine who I know is watching right now, Jacqueline. She and her
Thanks,
family
Jacqueline.
took care of me when I stayed with her family 3 times now when I've been to Brazil, and she had a woman working with her named Lady, and Leide, and Lady is she's just a special woman. She was the nanny to Jacqueline's children, and she took care of us when we went and visited in 2010. She made an orange cake every single day for my, or a banana cake. I'm gonna get in trouble if I get that one wrong. But she made a banana cake every day for my kids, and just an amazing woman. And she's getting a little bit older, and she's struggling with working. And so I wanted to do something to give back. And I was talking to Jacqueline about this last summer, and we came up with this idea that a little bit of money, a few $1,000s, can go a really long ways in Brazil. And Jacqueline lives in the famous favelas in a city called Maceió, and her house is literally falling down. Her walls were just not a condition that we in the Western world, or anybody with a bit of money would want to be a part of. You wouldn't think it was someplace you could live. And I knew with a little bit of resources and talking to friends and getting some donations that we could make a difference in her life. So we raised so far about $4,000, and we completely redid her entire inside of her house. We put a gate, we put a new roof, we've done everything what we call phase one, and she has a house that you can be proud of. So I would encourage, without taking too much time from this, I would love if people are interested in donating or learning more, or participating some way. Look on my Facebook, look up Ladies Home Project, and on Facebook, and participate, or just see the story. If you just want to see the story, it is amazing, the before and after. And Jacqueline in Brazil has spearheaded this project and gotten donations in Brazil, and it's awesome. And it's really heartbreaking because now the work's on hold because of the coronavirus. It's very bad in Brazil, as you can imagine. And so we're just hoping that they can make it through the rainy season, that the hot house literally is still standing at the end of all this. But right now, Lady's safe. She's in a safe place, and taken care of. She's actually taking care of Jacqueline's parents right now, so paying us back a little bit for the help. So
Wow.
yeah. So thanks for
Chad,
letting me talk about that, Shane.
Chad, that's a great story. And for those, the information is on Facebook, or
Yep.
give it, reach out to
Yeah.
Chad, or I can put you in touch
Please.
with Chad, whatever you're doing.
Yep.
Chad, Chad, we got Dr. Chad Briscoe here. Find him on
Yep.
Facebook, find out about Ladies Project. And it just sounds really awesome. And Chad, thanks for thinking of others. I mean, I
Yeah.
think about my journey as well. And so many people have helped me along the way. I owe so much gratitude. I'm
Yep.
grateful. I owe so much to so many
Yeah.
people. And so that's an awesome story, Chad.
Yeah.
And
Thanks.
we're coming up on the hour, and this
Yep.
has been a fantastic first show.
It's a lot
And
of fun.
it's
Yeah.
been really a lot of fun. I
Yeah.
hope it's been
Yeah.
helpful to everybody out in our communities. And so stay tuned. We're going to have another show on Wednesday where we're going to be talking with a pastor and a friend of mine. And he's going to be talking about the importance of faith through this COVID-19 and all the unknowns and all the
Cool.
uncertainties. And so
Yeah.
I'm super excited about that. And we've got a lot of other guests coming up for the next couple of weeks. We're kind of going to be focused on COVID-19 because that's what people are concerned about. And I want to get that information out to people. Chad, thank you for being a friend. Thank you for being the first guest. And I think episode 1 was an absolute success. I think it's even going to get better. So thanks, Chad. And we will be in touch. And thanks everybody for joining us. And we will see you soon. And never be outworked. Thank you.